Let's just jump right in, guys. Picture this. You've got one unsolved protein sequence. You need its 3D structure, and you literally need it right now. So today, we're taking that exact sequence and simultaneously firing it off to two of the absolute most powerful prediction servers on the planet.
Welcome to this explainer on the ultimate structure showdown, Swiss model versus I-TASSER. If you've ever predicted protein structures, you've definitely crossed paths with these computational giants. But, how do they actually differ under the hood? And more importantly, which one should be driving your next big project?
Let's look at the command centers. Over here is the highly streamlined Swiss model dashboard from XPassy. And right next to it, we've got the iTasser interface developed by the Zong Lab. We're going to watch these two just absolutely crunch the data.
Moving right along to section 1. One sequence, two models.
So what's the fundamental difference here? Well, Swiss model relies entirely on strict homology modeling. That basically means mapping your sequence directly onto a known template. iTasser, on the other hand, uses a really complex hybrid approach. It combines deep threading for fold recognition with ab initio fragment assembly to piece together the parts it just can't perfectly match up with.
Section 2. Swiss model, the speed king.
Honestly, just look at how ridiculously fast this pipeline is. It rapid fires a search through the protein data bank using BLAST and HH-Blitz. Then it automatically aligns the sequence, builds out the model using its ProMod 3 engine, and literally smooths out any physical clashes through energy minimization. Boom. Done.
But there's a catch. For Swiss model to really operate at its peak performance, you absolutely have to hit one crucial metric.
And that metric is 50% sequence identity. If your sequence shares more than 50% of its identity with an already known structure, Swiss model's automated pipeline is practically unbeatable. It's going to give you a highly reliable model in record time.
Which brings us to section 3, iTasser, the deep diver.
Now, iTasser is way more computationally intense. It starts by detecting multiple templates using Lomits. Then it takes those unaligned fragments and starts running replica exchange Monte Carlo simulations to assemble them. Finally, it uses SPIKR to cluster all those decoys and find the most native-like fold. It's a real heavy lifter.
So, the million-dollar question, when do you use which? It's all about knowing your sequence identity. We draw a really hard line here. Like we said, use Swiss model for closely related proteins above that 50% mark. But what happens when you drop down into the twilight zone? We're talking under 30% sequence identity. Standard homology modeling just breaks down here. And that is exactly where iTasser completely shines. Let's shift gears to section 4, assessing model quality. Because at the end of the day, how do we actually know if the model is any good? Well, both servers kind of grade their own homework, but using totally different metrics. Swiss model relies on GMQE and Q-mean scores. Meanwhile, iTasser validates its decoys using C-scores and TM scores. When you get your results back from Swiss model, this right here is the magic number you're looking for. A global model quality estimate, or GMQB, above 0.6. Since it's measured on a scale from 0 to 1, clearing that 0.6 hurdle means your Swiss model output is highly trustworthy and ready for biological use. Over on iTasser, the scale is completely different, so you need to keep your eyes peeled for this threshold instead. A C-score greater than -1.5, iTasser grades on a pretty wild scale from -5 up to +2. So if you manage to clear -1.5, it means the server has very likely nailed the correct global topology of your protein. And if we look at the RMSD measurements from the literature, it really proves a vital point. Homology modeling is undeniably accurate for close relatives, for sure. But iTasser's fold recognition is just brilliant at refining structural domains, even when those initial alignments totally fail. It basically pulls structure out of the chaos. Wrapping up with section 5, applications and final verdict. So, what does this all mean for your actual research? Well, these highly refined models are literally the foundation for massive breakthroughs in things like virtual drug screening and enzyme engineering. And here's a huge bonus. iTasser actually goes a step further by offering biological function predictions right out of the box. We're talking automatic annotations for ligand binding sites and gene ontology terms. It's a massive time saver. Which brings us to our definitive golden rule to take away today. Swiss model for speed, iTasser for depth. If you have a really clear template, just get it done fast and accurately with Swiss Model. But if you're stepping out into the unknown structural wilderness, let iTasser do the deep diving for you. So, with these two incredible computational powerhouses sitting right at your fingertips, what unsolved sequence are you going to fold next?